a whole genome annotation pipeline and prediction pipeline, and it annotates variants based off genomic elements, for example, known genes, location relative to gene splice site, etc., prediction of impact, coding impact, non-mass impact prediction, nucleotide conservation, linking elements, biological processes, biological pathways, prior knowledge, phenotype association, GWAS catalog, disease ontology, and many other databases. Also, it creates a advisor score. It's a score, a category from 1 to 5, 1 being pathogenic and 5 being benign, and it's based off ACMG categorization. And I will present a showcase today based on real case study, idiom study, patient number 9. Idiom study is idiopathic disease of man, research study with 6 causes of rare diseases, and it's based here at Scripps. Patient 9 is a 9-year-old European female, lifelong history of hypotonia, development delays, cognitive impairment, intermittent agitation. MRI shows subtle asymmetric volume loss in left hippocampus, motor milestones or delay. Onset of generalized tonic-clonic seizures was at 4.75 years of age. We did a quartet whole genome and exome sequencing. For whole exome variant summary, we had 101,797 SNPs and indels, 18,475 rare variants with less than 1% allele frequency, 1,053 non-synonymous truncating or splice site variants, 2 de novo inheritance variants, and 3 homozygous or compound heterozygous variants. To annotate, I will showcase how we annotated the whole exome variant summary variant and how we filtered and prioritized variants and came with a conclusion and found only one variant that is causative of this patient's disease. So we uploaded all of the VCF file into ASHG Advisor, which you can go on genomics.scripps.edu advisor. You can create an account to annotate file, but we also accept unregistered upload. I already have an account. I log in. We accept VCF files, tabbed, limited, bed-like format, complete genomics files, and to upload file, go upload file, choose file, desktop, I upload the VCF file. Depending on the file size, it might take a few seconds up to a few minutes. So right here in download results, it's processing, so the file is being annotated. I already annotated idiom file, so you can download the results by clicking on the download summary file. To analyze the summary file, you can go and use the ASHG Advisor UI, which is a tool, desktop application for that works on Mac, Unix, Windows computers, and it's the target of this application is for anyone, any scientist that might not have programming knowledge or doesn't know command line, to be able to filter and extract the variants of interest. So you can download the UI from here, and it's in download. Unzip it. Also, depending on the file size, you might need to change the heap size. We won't do this now, but on the web from the command on the web there are description of how you change the file size to upload up to 5 GB files. So for Mac, you double click on the command file, open, summary. So this is how the summary file looks like. You can see here gene annotation, gene type, non-coding RNA, protein coding, location, exon, intron. Further down, you see the allele frequency of 1,000 genomes, Welderly allele frequency, EQTL genes, splice site, protein impact prediction, polyphen, DB SNP, and advisor score is at the end. Disease ontology. Also, we have sorting functionality, custom filters. We have 13 custom filters. Statistics, you can calculate the summary statistics, save. At the end, you can save your reduced variants to a separate file. So, for example, coding and splice variant to frequency will extract all of the coding and splice variants that have allele frequency in 1,000 genomes below any threshold you insert here, or SG advisor Welderly allele frequency. There is a simple filter here. You can filter by any columns. You can enter your filter criteria. Advanced filter. Another filter that can be of interest, known disease filter, will filter only category 1 variants, advisor score category 1 variants. You might want to find out if any of those category 1 variants are homozygous or de novo in proband in the individual that has the disease. To do that, you load and compare the CF file, desktop, PTM9, progress bar tell you how long it will take, fairly fast. So we're back to the main filter. Once again, we do the custom filter, or the known disease filter, and you can see here the proband genotype. None of them are homozygous or de novo variants. This is the sister, mother, and father genotypes. To go back to the here, you can navigate through the previous filters that you created. So to go back to the original, show original data, you press Next, and you can filter. One interesting filter will be EDM filter. So you sort. The EDM filter will filter all of the de novo variants, homozygous variants, or compound heterozygous variants in proband only. So the father is unaffected, mother is unaffected, proband is affected, and we also have a sister here which is unaffected. We filter, and you see just a few variants. Some are homozygous, and some are de novo. This one is de novo, and this, after some manual literature search, we came to the conclusion that this variant is the disease causative variant. It's in polyphen. Right here, it's categorized as probably damaging, and we 2 other individuals were found with this variant. You can save this results to smile or to file. This as I said, my PI, Dr. Turkamani, found 12 individuals with variants in the gene KS and B1, and they were diagnosed with epileptic encephalopathy. And thank you for Dr. Turkamani, Philip Pham, and Bill Shipman.